our first speaker, Professor Ron Milo from the Weizmann Institute. Ron did his bachelor's in math and physics in the Hebrew University, then moved to the Weizmann Institute for a PhD in Uri Alon's lab, postdoc at Harvard, and then back in the Weizmann Institute, where he does, in my opinion, quite amazing work on various subjects, but I like most the look of numbers in biology, the census of the biomass of Earth recently, an amazing database that I used a lot of time, BioNumbers, that you could just search some value and then take out the number and more. And today he's going to talk about our very famous, recently famous bacteria that was all over the news, right? The carbon fixating E. coli. And with that, let's welcome Ron to the stage.
So actually, hi everybody, thanks for coming. So I didn't think about it before, but now that you mentioned BioNumbers, which is all about like having characteristic numbers in biology, I think there's also a strong relation for me between working on that and dealing with causality in biology, because we have a tendency, I think, in our minds often to have this dichotomy that either one thing is explaining something or a different thing is explaining something. And I find very often that like it's really important to follow the numbers, and then you get not a 0 and 1 or black and white, but to get a much more nuanced picture that enables you to get fractions and parts, and then causality becomes more of an interesting thing with numbers associated with that. So thanks for reminding me. And I think that could be a segue into the issue of causality and some things that we're doing. But as they say in Monty Python, now to something completely different. So we'll be talking about an effort in the lab of using E. coli to do carbon fixation. Let me tell you a bit about why we're doing it, what did it teach us, and maybe how it's related to causality. And so the world is happily divided in this kind of black-white picture by biologists into autotrophs, which are the things that know how to build a biomass directly from inorganic carbon dioxide that's in the air, and heterotrophs, those that consume them and depend on them. And we're a pretty good example of that. And the lunch that we just shared is an example of that happening. It's actually by a funny twist of things. I remember when I still studied biology in Kita Yud, like in 10th grade, that's the thing that caused me to decide I don't want to continue in biology, because the teacher was teaching us all about autotrophs, chemolithoautotrophs, phototrophs, heterotrophs. I said all this naming in biology doesn't mean anything to me. I want to move into physics and other stuff, and that's what I did. And in the funny twist of things, in the end, in the past 10 years, I've been working exactly on that specific subject. So life is full of surprises. And even though we study about these two things, in terms of like in a way you could frame what we did in the question of what causes a bacterium to be an autotroph rather than a heterotroph, or the other way around. And usually in our textbooks, it's sitting like in completely different sections. And that's something I suggest we go back to afterwards. Okay, and just how does that play in the big picture of things? So this is like a characterization of how the biosphere, as well as this conference, is being run. It's all sponsored by basically, energy, photons coming from the sun, and plants and some other bacteria, other organisms are pretty good at storing that for a short period of time by doing photochemistry of using that energy. And then you can't hold that energy for a long period of time. And then what evolution invented is a way to take those energy currencies, the ATP and the NADPH, and to store them in a good battery that does not require us to get all sorts of metals, rare metals from the earth. And that battery is made from sugars that are coming from CO2. And so this is the light reactions and the dark reactions of photosynthesis, and we'll be talking about those. But this process, even though it's very successful and drives the biosphere, also suffers from limitations in terms of the rate, at least from our perspective. We would have wanted it to work faster for the simple reason that there's 7.7 billion people on Earth, and we're quickly heading to 10 billion. So it's like a huge party that we're throwing, and it's not so easy to throw that party for a long period of time. And this issue of carbon fixation is at the heart of many of the resources that we're using of Earth. So in order to try and better understand it and explore it, we said, let's try and use a good organism to study that. And for that, we were thinking about E. coli. But there was a small bug in that plan. And that is the fact that the diet of E. coli is very much based on eating sugar and not on doing carbon fixation. And kind of like the question that we're asking is, what does it take and whether we have a chance to study carbon fixation in E. coli? And for that, we'll need to go through the first hurdle, a pretty significant one, would be to change the diet of E. coli such that it does carbon fixation. So basically, to reverse it such that it produces sugar from CO2 rather than the other way around. So far, so good? Okay, if I'm using any crazy jargon or something like that, let me know throughout. I don't want to understand just in the end that I missed you in the beginning. Okay, and this is the hero behind this work, Shmuel Gleiser. In this 10-year project I'll be talking about, he spent 7 years of his life and talent into trying and make this happen. And so we're talking, you remember in the big scheme of things, we're talking about what you see on your right, which is those dark reactions, the ugly sister of the light reactions in photosynthesis that has to do the dirty job of dealing with the CO2, etc., not about taking the photons and all that. Okay, so trying to do it in E. coli sounds a bit tough, but it could have been much worse. Why is that? Because if you look at how carbon fixation happens, there's this thing called the Kelvin-Benson cycle. There's like 13 genes within it. All of them are already within E. coli except for just 2 genes. This is Rubisco, the key enzyme that takes things from the inorganic world into the organic world. And then PRK that builds a substrate. All the other things are already part of the backbone of metabolism of E. coli. So in that respect, I'm saying it could have been worse. And so we'll still need to supply energy into the cell somehow. And that is being done in our case by using fomate. Fomate is a simple compound, C1 compound, that the cool thing with it is that E. coli could not assimilate it, so it could not grow on it, but it could achieve its reducing power, or energy if you like, directly from it. So it's a way to separate the issue of how to supply energy without supplying the carbon. At the same time, it's something that's being discussed a lot about a way to produce, you could produce fomate directly from electricity and CO2, so from air and renewable energy. So it could be something useful also in the future in that respect. I should clarify from the beginning, this fomate in supplying the energy turns it back into CO2. So it like comes with CO2 with like electricity and goes back into CO2. So overall, it's not that we have a solution to the CO2 problem currently, but I think it's an interesting proof of concept that could lead in those directions. Okay, so we spent a few years analyzing with all sorts of models that I won't get into on what else do you need to do in the rewiring of the metabolism of E. coli such that you'll have a chance in order to achieve that. And the short answer is that except for adding those 2 genes that I mentioned and another gene that would enable you to harvest the energy from fomate, you also need to delete some genes that would create all sorts of futile cycle, would basically lose all sorts of carbon into other reactions. So you do that, and that's kind of straightforward today in terms of like the ability to do with genetic engineering. Okay, so just to make sure that we're on the same page, what are we talking about? We're talking about, on the one hand, supplying energy with fomate and then going into a cycle. It's known as an autotrophic cycle. And that would enable you to build the biomass only from CO2. So we tried that. I told you what was the strategy. And I don't know if you'll be surprised or not, but it didn't succeed. And that was even though we tried for a long time and we're already deep within our ERC funding. So it was like not so easy to pull out of this. We had to find a different, like something to go forward with. And it was clear to us it's not so easy because we tried all sorts of rational things that we could think about what to delete, what to add in, how to play with the conditions. You do it, in the morning, in the evening, on a full moon, all the different versions, and none of them succeeded. So that was beyond our level of understanding. So we needed some expert thinker to help us. And for that, the only thing we could think about that could help us where we failed, given that we don't know what really needs to be corrected. Like it's in terms of the paper biochemistry, the rational design, it all makes sense. But in practice, there's all sorts of fine-tuning that probably needs to happen. For example, you need to play with the enzyme levels, maybe all sorts of things of regulation, maybe some black magic, all sorts of things that need to happen there in order for that to work properly. So for that, we decided to try and use evolution to help us. So here's the idea. This is what I told you that we wanted to have happening, and it didn't work. But we could start with something that's close by, but not the same. We could do exactly the design that we talked about, and we supply it with some surrogate carbon source or sugar source, like glucose if you want. It's called xylose, doesn't matter. It's a 5-carbon sugar, which the bacteria could survive on and even multiply, but we'll be supplying it at extremely low concentrations. Why? Because we want to put it under such conditions that on the one hand, it could still survive and do some adaptation through adaptive mutations. And at the same time, we don't want it to be super happy because we want to have strong selective pressure to evolve into actually using CO2. If you think about it, it's not so easy to achieve that, because if you're supplying very little of that, it would just eat it up and there wouldn't be enough to any more growth. So we're using this apparatus called a chemostat, which is exactly the kind of, it was like invented perfectly for our purposes. It's kind of like a torture machine for bacteria to keep them starved at a very, very low level. Anybody's interested, we could talk about how to derive the equations to show that. But the basic idea is you're putting bacteria that were designed there, you're putting them in very stressful conditions that they have at the same time lots of energy and CO2 available. And the question is whether they could evolve into actually living off of CO2. So far, so good? Yep.
Your ancestor is a regular E. coli?
Not anymore. It's an E. coli. It's starting from a regular E. coli where we added this rubisco, PRK, formate dehydrogenase. We deleted a few genes, and then it's our ancestor. Okay, so here, yep.
What about the interaction of rubisco with oxygen and not just with the CO2?
There is an interaction. We're working here at elevated CO2 levels and normal oxygen levels, and as you'll see, it is what it is.
So you don't have auto-respiration kind of?
There is some extent of photorespiration. That is one of the things happening here. Okay, so here's how the experiment goes. We're putting the bacteria inside it and we're following them. It's not a short experiment, it takes months. And then for a few, you could see here, after like 100 days, it's a bit hard to follow. Basically, sort of like nothing much happens. But then happily, after almost more than half a year, what we see is a decline in the sugar levels that we've put there. And at the same time, we see that the dependency on CO2 is becoming much stronger. What do I mean by that? We're basically now able, when we're taking cells out of the chemostats, we could now grow them on plates where there's only this formate and CO2. No need anymore for the surrogate carbon source, which is kind of positive, especially if you waited for more than a year now, because also some of them got contaminated, etc. But what can you infer from that? So one thing is, like, you could look at the growth rate, and you can see that really there is growth without any sugar that we've been adding in any form or style. But who knows, right? Like, we think that's what we did, but you might have something coming with the water, or maybe some of they're eating the plastic, or maybe something in the media, there's some contaminations. So you need to find a much more conclusive way if you want to go out to the world and tell them, hey, I have a bacterium that turned from a heterotroph into an autotroph.
The doubling time is 18 hours?
The doubling time is about 18 hours. Yeah, so it's also growing slowly. So you could say if it's growing slowly, maybe it's using something else. Maybe it's not really that. By the way, if you're an autotroph that needs to make a living out of CO2, that's not uncharacteristic. Like, if you're a cyanobacteria, some of them would be super happy at 18 hours. Okay, so might seem convincing, but all sorts of things could happen. How could we prove? And for that, we basically want to look at the air, be able to somehow color the air, and make sure to see if that color is what built the biomass. There's a nice trick to do that. You can use the fact that there's different isotopes of carbon. So you can put CO2 that's C13 labeled. So it's not radioisotopic, it's just a stable isotope of carbon. You're putting air or carbon dioxide that's labeled that way, and the question is what you'll see in the biomass. Okay, so basically you need to weigh the biomass, or not all the biomass, basically each and every component, to weigh and see those small differences of 1 Dalton or a few Daltons, depending on how many carbons it has. So for that, there's cool machines called mass spectrometry that enable you to achieve that. So this is like a map of metabolism. And you can see just in terms of inputs, we're putting CO2 that's fully labeled. And there's all sorts of parts there and amino acids. And basically in pink, it means that it's unlabeled, kind of like what we're giving it. And green would be fully labeled. And so the first experiment we're doing, we want to see that it's not eating something else. We gave it fully labeled carbon dioxide and fully labeled formate, also the energy compound. And as you might be able to see, it's like 99% labeled, basically fully labeled. So there's nothing coming from the plastic or from the water or something like that. We still want to make sure it's true that originally E. coli could not eat formate, could not assimilate formate, but maybe something has changed and maybe now it learned how to do that, which could also have been nice, but not what we were after. So you could do the same experiment now with naturally labeled CO2 and have the formate colored. And the question is, what would happen with the biomass? Okay, the idea is clear. So now you have the formate labeled in green. You do that and you see that everything is basically purple, so unlabeled. And then the final test would be fully labeled CO2, unlabeled formate. You do that and you see that indeed virtually all biomass is being built out of the carbon dioxide rather than formate or anything else. So basically this is an E. coli that is autotrophic. And if that does not excite you, nothing better is going to come after this, okay? Okay, so you could make the graphs and see exactly the values on that. And of course, what is really interesting now is to ask, okay, what happened in that almost a year when they were there inside the process of evolution, and what can we find in terms of what is causing this capability? So we did that. Basically, what we did is we sequenced everything that was evolving there, and we found that all the mutations, we had a few dozen mutations, and they basically could be decomposed into 3 different categories. One category is things that are related to the carbon fixation machinery itself, but not the genes themselves, but actually it turns out the bifurcation points, meaning the places where flux diverges out of the carbon fixation cycle into building the biomass. So on those gateways, we see that there are mutations. You can see a few of them here. So this one and this one and all of them. There's a second category, which are mutations that we find that are often happening in lab evolution experiments, kind of like the style that I was talking about, not trying to make E. coli autotrophic, but there's all sorts of lab evolution experiments that are trying to do things. And the third category of things that we don't even know what they're doing and makes no sense in a way. And either they're causative or maybe they're what's known as hitchhiker mutations, meaning because other mutations are pretty good, they just came for the ride with them because they were born or they occurred on a good together with good background. So how can we disentangle that in order to find what is causative, at least in our terms? So for that, I want to show you how do we find what is essential and how much can you play around with that. And that I'll show you those results. It's pretty tough to work with these bacteria at this stage. I'll show you on just the previous generation where we made the bacteria produce sugar but not fully autotrophic. It's kind of like a details. I think it's not so critical for our discussion. It's a very similar effort.
Can I ask you this one?
Yep.
So the ones that you have after a year, they still double slowly?
Yep.
Also 18 hours, right?
Yes. Yeah, you can push them with some other thing called turbidostat or just by serial dilutions to grow even faster. It's not so easy. Anyway, that wasn't really what we were.
I imagine that you keep following them, right?
We do.
And they keep eating CO2.
They keep on, yeah, because we're making them do that. We're just giving them CO2 and formate. We don't allow. If we give them now, say, glucose or xylose back, they're happy growing on that, and at some point they'll lose the capability to grow on CO2.
So in other words, you wean them completely off xylose.
Yes, okay. Indeed. Okay, so when we have a few dozen mutations, how do we go about understanding what is causative or not causative? I'll show you our version of doing it. So we're repeating the lab evolution experiment once again, because what we find is the mutations there, none of them is in the Rubisco or some other thing that we thought originally that would have the mutations. So we repeated this 3 times, and we did this Venn diagram that shows what is the intersection between the different things. And interestingly, we saw that there was one that was repeating in all cases, PRS. Never heard about it before. But now I had a good reason to become curious about it. And it turns out this is the main flux bifurcation point, the branch point, coming out of the Kelvin cycle. So this is a place where the cycle is really doing something useful, except for just taking CO2, by actually going and building biomass. So this is one of the things basically leading out, not coming in to the cycle. So then Uri, who's coming from was in the lab and is coming from a background of applied mathematics, quickly did the modeling of the equations of what does it take if you have a cycle, what's known as an autocatalytic cycle, which in a way it's like, the mystery of, life in general. We're all like an autocatalytic cycle that basically reproduces, etc. So there's all sorts of mathematical constraints on having such a cycle, such it would be stable, that it wouldn't run out of things just because there's fluxes coming out. So he was doing that analysis. There's stable points, unstable points. If you want, it's kind of like a bank account where you're getting some interest on the money that you have in the bank. And if you're withdrawing too much, you'll basically run out. So that's the condition for that when you're unstable.
So can you explain? So this was the good
I can, but I don't. I prefer to explain it to you rather than to everybody because it will take a while.
mathematically. Just why is this enzyme autocatalytic?
So the whole cycle, the whole cycle is autocatalytic in the sense that you're basically taking a substrate, which is organic, you're taking a CO2, Rubisco is the thing that merges them together, and basically you've made something organic that in the end you're recreating the substrate, but now you'll have more of that substrate. So basically what happens is that you do that 5 times on a 5-carbon sugar, and you're ending up with 6 copies of the substrate with 5-carbon sugars.
It's the intermediate, right? That it's
Yes.
Okay.
But the substrate is not the catalyst, so it's not autocatalytic.
It's something that accelerates
The combination of the substrate and the enzymes together is autocatalytic. The combination of, it's an autocatalytic cycle. Okay, it's not that the enzyme is autocatalytic, it's not that the substrate is autocatalytic. The combination of the different intermediates together with the set of enzymes, all together, it's an autocatalytic cycle, autocatalytic system.
Okay, new definition of autocatalytic.
Okay, so we could talk about it. Maybe I'm aware of that. I'm aware there's different definitions of different hierarchy, levels of hierarchy, but it sounds to me like this is for the coffee break. Okay, so there are conditions on stability of that, and I guess I'm showing you that because it goes into some biochemical details, but in the sense of what is causality for us, this is sort of like in that direction for us when we want to go deeper. So you can do that analysis, and basically you make a prediction that what needs to happen is something about the ratio of the rate, the Vmax versus the affinity, and there's some condition, and so we could check on that. We're taking and finding where are the mutations in each of, I told you it appears 3 times, right, in this Venn diagram. It turns out that every time it was in a different locations in the enzyme, so it's not the same mutation, it's the same gene, but not the same mutation. We could map that on the structure of the enzyme. It's these things that you see there in red, and you can see that it's somewhere next to the catalytic pocket, but not inside the catalytic pocket, which sort of like makes sense to us, but who knows, because like it makes sense to us in the sense that you can't ruin the activity, because then you can't build biomass. But you have to change the activity, because right now it's used to getting glucose and using it happily. Now it needs to be much more sophisticated. So it needs to, basically it's a dial that we think needs to be tuned down. At least that's what the modeling tells us. And now we see that, we could make some hand waving, or my friends that know structural biology probably could make some hand waving on that. How can we sort of like prove that even further? We're doing the biochemical assays where we're comparing between the wild type. So what you see here is as a function of substrate, what's the specific activity, what's the rate at which it's working. There's the original one. And then we're seeing that all 3 independent things that were happening all decreased the activity, which is in line with what we understand about the need for the stability of the autocatalytic cycles.
And if you make double or triple mutants?
So if we make a mutant such that it, you mean of both things together?
AGPT and G22.
We didn't try that. We didn't try that.
That wouldn't be very sensible because they'd be too low.
Maybe.
Or not.
The short answer is we didn't try that. I know that when we're deleting it all together, it doesn't work. Yeah. When it's a wild type, it doesn't work. I know this worked, but let me show you what we did, which is sort of like there's a lot to be done here, but I'll be happy if somebody does it, if not us. Okay, so how can we understand? So this was nice about this one specific example, but now I'm telling you we have like 20 or 30 mutations happening. It would be super tough for us to do this analysis for each one of them. So what we could do is basically we want to make a more compact set of mutations to find like the minimal set of essential mutations. So what we did, we already know that this is essential, so we're putting that in. And now we're just repeating the evolution again. And then it happens sort of faster. And what is even more cool that now, except for that now happening after a month or two, now there's only 8 mutations, so not 20 or 30 mutations. Now it's smaller to a set of 8. And we could look at that 8. We could actually even see that some of them we probably don't need, so we can remove those. And we're left with an even smaller set. Okay, I guess this might be the smaller set. Yeah, 1, 2, 3, 4, 5 mutations. Okay, what are these 5 mutations? 3 mutations, which are all these bifurcation points, those branching points from the Kelvin cycle. And one that is related to regulation of energy, and one which is this global regulator that affects all sorts of things. Okay, so now we're so like in our causality, aim to get at causality, we have like a smaller set, still not so easy to understand. So the question we could ask now is, when we have a mutation, say in SER-A or in PGI or whatever, if we were repeating, let's say we just remove that, and we're asking evolution to solve with the other mutations, but just to have an extra one mutation, would it choose the same target, or it could have all sorts of different options? Let me just 1 second. Okay. So that was the question we tried to answer. And there's a cool trick for that, because what you can do is you can remove one of the mutations, then the cells could not live in those stringent conditions. But you can have a big population grow under not stringent conditions, but like in happy media where they have everything they want, glucose, whatever. Then they multiply to a big amount. Actually, the number is so large and the mutation rate in bacteria is such, basically you'll have every possible mutation in the genome happening in at least one bacteria. And then you can take those, you can plate them now on stringent conditions. Okay, so listen what happened. Okay, I brought back one of the mutations, so this is not good. But now I build a big community of bacteria that each one has a separate, different mutation. Now I'll be plating them again, and I'll be look for the few colonies, even though I'll put like 1 billion bacteria, there'll be like 5 colonies, which have mutations that turn out to be able to be good solutions to the puzzle of something that could solve this issue. And the question is whether it would be the same one, it would be something else, the same mutation, the same gene. That's the question we were asking. And so this is the final thing I want to show you. So this is the work of Elad Hertz in the lab. So he takes, for example, this PGI. This notation says we brought it back to the wild type. We grew up, billions of bacteria. We put them back, we plated them on a selection media now that they do need to be able to use that. And we were looking at what are the mutations that were able to do what biologists like to call complementation. And so it turns out for PGI, what you see here in stars is the places where we find colonies that have mutations. So it turns out in 5 cases, we had mutations in the same gene in PGI. And in another 6 cases, we had the mutations not in PGI, but in PGM, which is the next gene downstream in the pathway. So what does it tell me? It tells me it's not that you necessarily have to have that gene, but it seems like evolution, the target is within that biosynthetic pathway. So it's either this or that. Okay, given the background that we're standing in. You can do that, and it makes sense to me because if we're saying you have to tune down the dial, you can either do it here, or it's also okay if you could do it here. Okay, you repeat that the same thing in another branch point, this thing related to serine production. You repeat that, and there you find that several times, indeed, you have the mutation again there. And in some other cases, it seems like you're tuning down a different dial, but maybe further, I'm not sure. That's the story we're making now because we didn't analyze it deeply enough. But it seems like there could be an alternative, but it's still pretty much close to where you started with. You can do it also the global regulator, the CRP, and then we're finding some interesting things. Either it's in CRP or in something else that in this complex genetic networks that you can do some other thing, that's something going up, something going down. It's quite detailed. And I guess just in ending, I want to say there's this nice statement by Orgel that evolution is smarter than you are. I think this work shows you in all sorts of different ways how it has a lot of tricks that it could teach us. But I think going together with that at the same time, we could also become smarter through evolution. So I want to share with you some of the things that I felt like that we learned from this. So one thing is about the immense power that selection has. So if you need to reach a goal and there's a way to use evolution for that, selection is a very potent tool in order to enable that. So I think you saw it in different ways, and chemostats are good tools for the kind of processes we were talking about, and you usually need this combination between rational design and lab evolution. So this hybrid is especially potent. The other thing that I think we've learned is that metabolism is highly plastic. So if I go back to this issue of like, what does it take to move from, heterotroph to autotroph, even though it's two completely different sections in the book, nature doesn't care so much about how we structured our textbooks. It turns out that within a few weeks or months in the lab, it could move from one chapter to a completely different chapter. Everything is connected in the space of metabolism. It has a lot of plasticity to change from one mode of growth or one diet to a very, very different diet. And finally, if you want to elicit such changes, it seems that these branch points are at the heart. It's like a design principle, if you like, of what does it take to change an autocatalytic cycle. Okay, so I showed you about how we started with a heterotrophic E. coli, the standard one. We used engineering, which did not help us enough. Then we used also lab evolution. This brought us to an autotrophic E. coli. And I hope that in some effort of many, many other labs around the world that, as it works in the world of science, we publish this. Hopefully, people will take it further. I think with future work, you can give such bacteria maybe alternative energy sources, renewable electricity, etc. And hopefully such bacteria, which are workhorses in the biotechnology industry, could help us in the big challenge of how do we deal with 7.7 billion people heading to 10 billion. That's about what I wanted to tell you. We're looking at also at like playing around with the CO2 levels, understanding the genetic basis even further. In terms of issues of contamination to the environment and things like that, it's something that is also on our mind, whether such bacteria could be harmful in some way to the ecology and something like that. So what we're all the time doing is like we're asking people as well as ourselves to see if there's any scenarios that they could think about, which is rational, not just fear by itself, but like something that has a logic to it that could be a worry. And we wrote a document documenting any such worries. And for each one, I think we have very good explanation why this is not something that could materialize in any possible way that we could think about. But if you see something differently, please let me know. That's about it. I guess except for Shmuel, I want to especially mention the work of Ro'i, which is working on this, Melina and Eyal, together with Inon, some other members of the group. And this has been like 11 years in the making. So I guess I should thank Aaron Bar-Even, who brought me on this project as a brilliant PhD student, had the crazy idea of doing that, together with Elad Noor that did the modeling to see how we could make that work. And this was all helped by the ERC that were supporting it for a long period of time. And Shira, that you can see here, started with doing those lab evolution that I told you about as like an undergrad coming to work in the lab. So she just did it manually. It did not work, but she continued in other things. She did her master's in the lab. And just to show you, this is like a few years later, this is in her wedding. She's not in the lab anymore. Now she has a kid, which is a few years old. And now we finished this project. So that's kind of like the time scale that it takes as well. Thank you very much.
I'll take the microphone. And do we have any questions? Yes.
In the last set of experiments you showed us, you could revert one of the mutant genes back to the wild type, and then you select it once again under stringent conditions. Might some of the alleles, the new alleles arising under stringent conditions, be even better in terms of reducing the doubling time of the bacteria, which seem to be a major obstacle in making this more effective in terms of reducing the doubling time to a matter of several hours rather than a whole day? Might some of these alleles be even better in accelerating the doubling?
They might have. We did not spend time at characterizing that. That's the short answer. Our aim was sort of like as a proof of concept that we could do this. We weren't looking to optimize the growth rate. There could be all sorts of ways. This could be one way to optimize the growth rate. There was a different suggestion before about how to optimize the growth rate. This could definitely be a direction to go.
It's surprising. It's surprising there has not been the outgrowth of more rapidly growing subpopulations of bacteria, given the mutability of the E. coli genome, because there would be a very strong selective advantage of being able to grow faster, I would imagine.
Yes, so I think we might have a limitation in how, having some experience in microbiology, we tend to think about 18-hour doubling time as something super slow for an E. coli, but in a way, this is not E. coli anymore, right? It's like an autotrophic organism. Autotrophic organism, usually none of them grows at 20 minutes. None of them grows at 1 hour. So 18 hours is very characteristic. It's not so clear how easy it is. You can just do also the calculation of what it takes in terms of resource allocation for Rubisco, for PRK, for form-A dehydrogenase. It's a significant fraction of the proteome. Evolution was trying for many years to push things like Rubisco to be faster. It's not so easy. The one thing that we do find that helps us, it relates to a question that was mentioned here before about photorespiration, the interaction with oxygen. We do see that when we're decreasing the level of oxygen for, say, 20%, which is the ambient thing in the atmosphere, to about 5% that we could easily do in the lab, they do grow faster. I don't even have the exact number off the top of my head, but they do grow faster, and that makes our life somewhat easier to work with them. That's what I could say.
Mammalian cells also grow better at 5% than 21%.
I didn't know that.
Yeah.
Yeah, they do?
Oxygen's toxic.
I didn't know that.
Yeah. I didn't quite remember or understand how many mutations you need in order to convert these bacteria to become autotroph. I mean, in nature, autotrophy and heterotrophy are not so far apart as in textbooks, because plants in the night, they behave as heterotrophs. And so maybe many mechanisms are shared in these two kind of metabolism, and that's why you can convert them so easily.
Yes, I'm not sure that I would call plants at night. It's true that they're using the glycogen pool or the starch pool, usually the starch that they've been accumulating during the day. But let's say doing the transition, like you could have autotrophs that then are using that sugar that they accumulated to live off of that in the reverse direction, which is what we're talking about, something that's a heterotroph, to become an autotroph. Well, in a way, probably everything started at some point as an autotroph some billion years ago. So in a way, the E. coli is coming back to something that it probably was, I don't know, many hundreds of millions of years ago. But the fact that this transition does not require so many genes, to me it was a surprise. I have to say that, after I talked about the idea of this project, with biologists and gave talks about it for quite a few years, everybody basically thought it would never succeed. So it's not like something that was clear it's going to be just, a few mutations that will get there.
Craig Bettner believes you can design an E. coli from scratch. I believe you can make a synthetic E. coli. Have you considered doing that? I mean, basically, you start with DNA and just design the whole genome.
So we didn't consider that. It's not my thing, I guess. But I would also say when he says from scratch, I think what he talks about is just this technical aspect of the fact that he could synthesize a long enough stretch of DNA so that he could boot a cell with that DNA that was synthesized from scratch. I wouldn't call it that like he could make a bacterium from scratch or an E. coli from scratch. like, it's not that he knows how to make a bacterium, you know, from basic principles.
He wants to know the minimal number of genes
Oh,
that
yeah.
would work in that organism, or indeed simpler organisms.
Yeah, so that as well, I would say it's not really from scratch. It's like, you know,
No,
it's
yeah.
like taking so much, you know, information from what evolution has done and then reassembling like a compact set. So I guess it's not unlike what we tried is a very, very small version of that in terms of the mutations that led, can we find a compact set? So that's, you know, very alluring in terms of like an elegant thing to do. You know, we haven't been thinking about such things.
But I don't know whether he has considered or knows about your work, because you've elegantly shown, sorry, I don't know if people can hear me, you've elegantly shown that the subtle control mechanisms are necessary to accommodate different genes by showing that you need to fine-tune. Your mutations show you need to fine-tune particular enzymes in order to achieve what you want. So in a way you are redesigning the E. coli, aren't you?
Yeah, there is.
You are redesigning.
There is a redesign here. There was like a basic science question of whether you can do that transformation, and there's a follow-up question whether you could use that for the benefit of humanity.
So I don't know if he succeeded in his experiments. Maybe Bob Weinberg will know.
So-
Absolutely not.
He hasn't succeeded.
Bob
I mean, he's a
Weinberg
great
has no
talk, and
idea.
I don't
I
know
know.
what's
That's no
happened.
idea.
So I would say.
No idea.
So I've been following on that a bit. So you know, it's like there's a success in every step in a way by definition. Basically, he tries to minimize it, and every time it's a bit smaller, it's a success. And whether it could be still smaller is probably you could still do it smaller. But I would just say, in a way, in a way, what we're doing is like basically we try to see if we could evolve the bacteria to live on much harsher conditions.
No, I like
And,
your
no, I'm not from a defensive point. I just want to point it out. What he's trying, like the way to achieve this minimal bacteria that he's after, is by putting it under conditions that are as ideal and optimal as possible. So not only would they not need to be autotrophic, basically let's supply them with the richest possible media, everything already preformed, and let's see what are the remaining things that still get rid of as much of metabolism as possible, as much of the control as possible. So it's in that spirit, I think, that effort is moving.
And we have time for a last question.
Hi. I have a question, like two questions actually. I'm gonna do it quickly.
Let's have three, and I'll be like the genie in Aladdin, like three
No,
requests.
I don't want to be greedy. Evolution is a multidimensional phenomenon, you could say. So, do you think you can do evolution in like multiple conditions simultaneously, like push it in few directions at the same time? That's the first question. And the second question.
Can I answer that because I might forget afterwards?
Sure.
I think in a way it's happening without us even wanting. Like we thinking that we're doing evolution to be autotrophic, but we don't really understand the psychology of the bacteria. It probably, you know, the thing that it sees the most is like it's going in these chemostat conditions, which is very strange condition. So it's probably adapting to that and to this limitation of so there's several things happening here concurrently. In that respect, we also didn't know what would be the outcome. It might have all sorts of, you know, it might just do that and not what we were looking for. Yeah. Happily, this and that.
Okay. And the second question, I think in your paper you said like accumulatively it took you 6 months to do all the mutations, if I remember correctly.
In the chemostat. Yeah. So the first indication that we had in the chemostat of these bacteria was after half a year.
Yeah. So do you think if you like insert a mutagen and like accelerate the mutation rate, it might take shorter times?
So we tried doing that and then it didn't help. In some other experiments, we did see that there were cases where it happens naturally and it still takes roughly the same amount of time. And if you want, we could talk about I think we have some understanding of the population genetics behind it, why I think it wouldn't be super helpful, but we'll see. It's not that we fully understand that. What's sure is that when you do that, then it becomes a huge nightmare to try and understand causality, because instead of like 20 mutations, you're having 600 mutations, and then go figure.
Okay, let's thank Milo again for a great talk.